So thank you all for joining us for the first Science Cafe of the spring semester of 2021. I'm very happy to introduce our speaker today, who's Alisa King-Klemperer. She did her undergraduate degree at the University of Wisconsin-Madison, and then she went on to do her PhD, her master's and her PhD work here at UIUC. And from there, she came and joined the IGB and started her career and got a job at the IGB in science writing. And she's going to talk to us a little bit about her research and her journey to her career in science writing. So I'm very excited to hear her talk, and go ahead and take it away, Alisa.
All right. Can everybody hear me? Okay. All right. Thank you, first of all, Danny, for organizing this and inviting me to kick off the first Science Cafe of the semester. I'm really excited to share my story with everyone today. My story is going to take you on a journey from my PhD career to my current profession, which is a science writer. So, like Danny mentioned in the beginning, my journey began back in Madison, Wisconsin. I was actually interested in microbes or microorganisms ever since high school. A freshman biology class sparked my interest in the subject. And ever since high school, I knew I wanted to study microbes. I obtained my bachelor's degree at UW-Madison and was told of a very prestigious microbiology program at the University of Illinois, Urbana-Champaign. With that, I decided to apply to the PhD program at Urbana-Champaign and applied to the microbiology department within the molecular and cellular biology umbrella program. I obtained my master's degree along the way, and after a few years, I completed my PhD in microbiology. And in that same year, I started my job as a science writer fellow at the Carl L. Woese Institute for Genomic Biology, or IGB for short, which you'll hear me say at the end. So my talk will be split into two halves. So the first half of my talk will be talking about my PhD work, so more sciencey, more technical. And then the second half of my talk will be transitioning into my career and how I decided to get into science writing. So for my first part of the talk, I will be sharing part of my dissertation work, which once again does not encompass all of my dissertation. Specifically, I will be describing how we employed a combinatorial approach to identify small RNA target predictions for both well-studied and poorly characterized small RNAs in the bacterium E. coli. So why do we care about microbes? Why do researchers study microbes in the first place? Well, microbes are everywhere, and as a result, they play a very important role in all of our everyday lives, with some being responsible for a significant proportion of diseases in not only humans, but animals and plants. Microbes are also important for modification of the environment, such as the water and soil and forests. Scientists have learned to exploit these microbes in the field of molecular and cellular biology, and as a result has led to engineered solutions, such as the production of beverages, food, and antibiotics that provide lots of benefits medically and industrially. My research falls into the fundamental microbial sciences category, where the research that we conduct gains insight into the microbial processes. For my PhD, I had the pleasure of working in the Vanderpool lab under my former advisor, Dr. Carrie Vanderpool. here in the Department of Microbiology on campus. Research in the Vanderpool lab focuses on small RNA regulatory networks and defining the molecular underpinnings of RNA-mediated regulation in bacteria. Other interests include bacterial physiology and stress responses, and most recently, the gut microbiome. Before I dive into my research and talk about some data, I wanted to explain very quickly what the central dogma of molecular biology is. So the central dogma of molecular biology describes the flow of genetic information from DNA to mRNA to protein. The central dogma of biology states that the genes encode the sequences, specify the sequences of the mRNA, which in turn specifies the sequence of protein. We can use an analogy to understand this process better. So for the DNA, you can think of it as a blueprint for a building, for example. The blueprint contains all the information necessary to construct the building. You can think of it as a critical piece of information that is critical for the cell. The blueprint can then be photocopied, so many copies of this blueprint can be made. And the process of taking DNA and making multiple copies is called transcription. And so DNA is transcribed to different RNA species, such as rRNA, tRNAs, and sRNAs. mRNAs is what I will focus on for this analogy. Next, you have your photocopies, and those photocopies can then be made into the building. So you have your construction workers, which act as the ribosomes. And ribosomes, you can think of ribosomes as a factory. So they're the ones that are churning out the protein. The process of taking mRNA and making protein is called translation. So essentially, the construction workers take the blueprint and you get the final product, which is the building. So my research specifically focuses on what happens post-transcription. So after DNA is converted into RNA. I won't be talking about the other species, but I will be talking about small RNAs for my research. Like us, bacteria live in rapidly changing environments and constantly deal with a bunch of stressors, which are depicted as lightning bolts here in this figure. Some of these stressors include toxins, nutrient limitation, and pH stress. In order to succeed, bacteria have adapted the ability to respond to these signals by altering gene expression. This gene expression can take place in transcription, where you have repression of transcription, so preventing those photocopies from being made, or prevention of translation, so preventing a protein production. By changing gene expression, the cell's properties and behaviors are altered in response to the stress signal. Regulatory RNAs, such as small RNAs, are responsible for regulating mRNAs. So they are responsible for post-transcriptional regulation. So small RNAs are found in hundreds of bacterial species, with over 300 small RNAs identified in E. coli alone. There are about 2,800 interactions that have already been characterized for 70 E. coli small RNAs. Although a handful of small RNAs have been discovered, verification and identification of targets is only known for a small dozen of small RNAs. This verification is the rate-limiting step in characterization of small RNAs. In a lot of small RNAs, we still don't know what the physiological function is. So a little bit more about small RNAs. Small RNAs are usually 50 to 530 bases in length and are non-coding, meaning that they do not contain instructions to make protein. Small RNAs usually contain a single-stranded region, shown here, and a seed region that base pairs or interacts with its mRNA target, shown at the bottom. This interaction, the base pairing interaction between the small RNA and the mRNA, is called the seed pairing region. RNA chaperones bind and stabilize the small RNAs and mRNAs and promote base pairing interactions. So next, I'm going to walk you through small RNA regulation, and most of the regulation is either positive or negative regulation via modification of translation initiation or mRNA stability. The first example is a case where you have positive regulation. For positive regulation, you have an example where binding of the small RNA to an mRNA opens up an inhibitory structure that allows the ribosome, so those factories that I showed you, to come in and make protein from the mRNA. So this positive regulation allows translation to occur. Another case of positive regulation is the same, except binding of the small RNA to the mRNA protects the mRNA from degradation. However, most cases of small RNA-mRNA interactions involve negative regulation. Here, the small RNA binding to the mRNA occludes the binding site of the ribosome, preventing translation from initiating. Lastly, there are cases where repression of translation is also coupled to degradation of the mRNA. So you learned a little bit about small RNAs, but why do we study small RNAs? Why is it important to study them specifically in bacteria? Well, first of all, discovering small RNAs and characterizing small RNAs has solved long-standing mysteries in bacterial physiology. Through small RNA discovery, previously unknown pathways and metabolic processes have been discovered. Lastly, studying small RNAs has revealed new molecular mechanisms of RNA-mediated regulation. So here I'm just showing a regulatory network. So what you have here in the big colored circles are examples of well-characterized small RNAs. So for example, here is a small RNA, RIB, which I will talk about later. And from each small RNA, you see little lines leading out to nodes. And these nodes represent the targets that they have been characterized to regulate. For example, the targets that RIB has been shown to regulate are depicted as little green circles. We call the collection of target genes regulated by a small RNA a regulon. So this would be the RIB regulon. Since small RNAs have multiple targets, these connections can be overlapping and interconnected, once again allowing us to reveal unknown pathways between processes. However, we still knew very little about some of the small RNAs and their regulons. So this was kind of where I came in to my PhD work. We knew that hundreds of small RNAs were being discovered, but we realized that there still were targets to be identified. And the rate limiting step was in fact target identification of small RNAs. Currently, there exist both computational and experimental methods to identify mRNA targets. Computational methods can produce a lot of putative targets, but they also have a high false positive rate. On the other hand, experimental methods might provide more direct evidence of an interaction, but they can also be labor intensive. Since there isn't one fault-proof way to identify small RNA targets, we hypothesized that employing a combinatorial approach that encompasses both computational and experimental pieces of evidence would improve predictability of bona fide targets. To tackle this, in collaboration with Dr. Patrick Degnan at UC Riverside, we developed a pipeline called sRNA Target Prediction Organizing Tool, or SPOT for short, which streamlines the process of computational predictions for bacterial small RNAs. In short, SPOT is able to run 4 publicly available algorithms shown here. And these algorithms basically work by scanning the genome and looking for base pair interactions between the small RNA of interest and mRNAs. To use SPOT, the user provides a reference genome from any species and the sequence of the small RNA of interest. The user can also change parameters however they like, and with these, SPOT runs all 4 algorithms in parallel. SPOT is also able to collate the results from these 4 algorithms. In addition, experimental evidence can be integrated and results can be filtered to produce an experimentally tractable list of small RNA mRNA interactions. So there's a lot going on in this figure, but first we wanted to test SPOT and see how robust it was. And to look at robustness, we looked at the measure of sensitivity. And sensitivity here is defined as the number of correctly predicted targets over total known targets. So what we mean by known targets are targets that were experimentally validated through other studies. And so here on the x-axis, we have a number of small RNAs that were previously characterized in other studies. On the left y-axis, we have the number of predictions. So you can think of this as the number of targets that were predicted for that small RNA, with each bar representing the number of predictions for those small RNAs. On the right y-axis, you have the true positive rate or sensitivity. And once again, sensitivity is defined as the number of correctly predicted targets over known targets. So the higher the sensitivity, the better. If we just focus on the red bars indicate targets or genes that were correctly predicted by 2 or more algorithms. And you can see that a lot of the known targets were predicted by at least 2 or more targets, looking at the height of the red bars. Now, if we look at SPOT, here depicted by black diamonds, we tested SPOT and saw that the sensitivity of SPOT was greater than 75% across all small RNAs that we tested. All in all, combined evidence gives a higher likelihood of correctly identifying a target. Based on these results, we saw that SPOT achieves greater than 75% sensitivity and less than 50% false positive rate when at least 2 methods converged on a prediction. So once we tested SPOT on some well-characterized small RNAs that we knew the targets for, we wanted to go ahead and extend this analysis on well-characterized small RNAs, but this time looking for new putative targets. So in order to narrow down the list of putative targets further, we came up with a pyramid ranking scheme shown here. And this ranking scheme incorporated predictions from SPOT in addition to experimental evidence, either from other studies or from studies conducted in our own lab. For the pyramid scheme, we have 4 tiers, with the bottom tier at the top being the least stringent, meaning that these targets that were predicted were only computationally predicted, landed in tier 4. As we go lower down the pyramid or higher up, whichever way you see it, the parameters become more stringent, meaning more pieces of evidence are required for the likelihood that it is a bona fide target. So you can imagine targets that are in the tier 1 are pretty likely to be a true target because they combine both computational and multiple pieces of experimental evidence. Given this ranking system, our hypothesis was that the higher tiers with more stringent parameters would yield less putative targets, but have a higher confidence it's a true target. So for each small RNA, I ranked the predicted targets and went ahead and constructed fusions to test for regulation. And as a reminder, I am looking for post-transcriptional regulation. So keeping in mind the predicted base pairing interaction, I went ahead and constructed these fusions. And I only paid attention to those that fell into the top 3 tiers. Then I went ahead and overexpressed the small RNA of interest in those backgrounds and performed assays to confirm or neither confirm or those regulation of the small RNA on the mRNA. So for this part of my work, we looked at 4 well-studied small RNAs, but tonight I'm only showing 2 of the small RNAs. We looked at RIB and GCVB, and both involved in regulation. So RIB is involved in iron homeostasis within the cell, while GCVB is involved in regulation of amino acid uptake and biosynthesis. Again, our goal was to capture new putative targets for these well-characterized small RNAs. So before I get into the data for GCVB and RIB, I just want to quickly explain why amino acids and iron are essential in bacteria. Amino acids in bacteria serve as the building blocks for protein, and these proteins are involved in many processes within the cell. As for iron, it is used to coordinate gene expression, facilitate protein enzyme function, and in some cases, aid in pathogenesis. In the case of RIB, RIB is involved in iron homeostasis within the cell. Under iron replete conditions, expression of RIB is repressed, and these genes that encode proteins involved in these categories are allowed to be expressed. However, in iron starvation conditions, RIB expression is no longer repressed, and RIB can go on to regulate its targets. This is a way for the cell to control the iron pools, especially when the cell is iron starved, and to make sure it's not expending energy or iron unnecessarily. On the other hand, GCVB is known to be involved in regulation of genes that encode proteins involved in amino acid transport, metabolism, and biosynthesis. With one of the biggest regulons to date, we were particularly interested to see if we could identify any new targets for GCVB. So this table has a lot going on, so I'll slowly walk you through this table. but this is the table that I came up with after I ran SPOT and also after looking at experimental evidence, other studies or my studies in the lab. So we have GCVB and RIB here on the left, and each column represents one of the tiers of the pyramid scheme I showed earlier. Here I'm showing two different numbers. So the numbers in black represent predicted known targets. So once again, known meaning that these targets were already experimentally validated. In the red, we have predicted unknown targets. So these targets were predicted, but did not contain experimental evidence. And as you can see, there are a lot of targets that were predicted that fell into the tier 4 category, meaning that they were computationally predicted. On the other hand, you also see that both GCVB and RIB had a high number of known targets represented in tier 1. Next, from this table, I wanted to focus on the unknown targets that were predicted. So looking at the red numbers. And so I decided to go ahead and test the targets that were predicted in the top 3 tiers. So here I'm showing an assay that I conducted looking at regulation. And so here at the bottom, you have the target genes that I tested. So those targets that fell into the top 2 tiers. And here we are looking at GCVB. We split the targets into 2 tiers. So tier 1 targets and tier 2 targets. On the y-axis is the measure of activity that I used to look for significant activation or repression of the target by the small RNA. The green bars represent overexpression of the small RNA, while the blue bars represent no expression of the small RNA and therefore acted as a negative control. When I tested all these targets, making sure to include a control a target that I knew was regulated, I saw that 8 targets were significantly repressed, with 1 exception being activated by GCVB. And so from this, we concluded that GCVB directly regulates these targets. If we go back to that previous figure and we superimpose the new putative targets, we found that these targets fell in line with previously known targets, meaning that the targets that we identified encode proteins that are involved in similar functions as other targets in its regulon. So next, I conducted the same analysis, but this time for the small RNA RIDC. And so here I'm showing the targets that I tested that fell into the top 2 tiers and another control. So this target here was already shown to be regulated by RIB. From this, I saw that 3 targets were activated and 1 repressed by RIB. And once again, from this, we concluded that RIB directly regulated these targets. This time, if we look at the RIB figure that I showed earlier, we see that these target genes encode proteins that fall into the iron-using protein category. This makes sense if you think about it, because if you remember, RIB is repressed when iron is abundant, but activated when iron is starved. So you would not want proteins using up the iron if you are starved for iron. And so it makes sense to me, it made sense to me that RIB repressed those genes. From this part of my work, we were able to successfully identify new targets for the well-characterized small RNAs, GCVB and RIB, using this combinatorial approach. We also saw that sensitivity was greater than 75% for both RIB and GCVB. Lastly, we decided to extend our analysis to a poorly characterized small RNA. Shown here is RIDC. As you can see, RIDC is a relatively small RNA, only 60 nucleotides in length. At the time when I started working on this project, not much was known about RIDC, besides the fact that overexpression of RIDC caused growth defects when E. coli was grown on different carbon sources. However, later on, my former colleague, Colleen Bianco, was able to characterize RIDC further and found that RIDC was important for acid resistance. So once again, I conducted the same analysis for RIDC. And so I used a ranking scheme to order the targets, both predicted known and unknown, to see if I could test any additional putative targets. Compared to GCVB and RIB, RIDC has a much smaller regulon, so the numbers are a little smaller here. Once again, there are a lot of targets that were computationally predicted, but I decided not to test those. For my analysis, I focused on targets that were predicted and landed in the top 3 tiers. And so from this, I conducted the same analysis. So if we look at the analysis for RIDC, I saw that 2 genes are regulated. So the first gene, Phe, was repressed significantly, and another gene, TRPE, was activated. Looking closely at both Phe and TRPE, we see that they encode gene proteins that are involved in amino acid biosynthesis. Phe is involved in phenylalanine biosynthesis, while TRPE is involved in tryptophan biosynthesis. It was interesting to me that RIDC was able to repress one and activate the other, and they both use the same intermediate. This could be a way for the cell to modulate the biosynthetic pools of these amino acids. So conclusions for part of my thesis work. First one, there was a higher confidence interval for true targets when multiple pieces of evidence converged on a prediction. We were able to expand the target regulons for poorly characterized small RNA RIDC and well-characterized small RNAs GCVB and RIB. Finally, the employment of a combinatorial approach accelerated the rate-limiting step of siRNA characterization, which is target identification. Okay, so now I'm going to switch gears and talk and transition into how I decided to become a science writer and what steps I took leading up to that. So before I get into my career, I have to go all the way back to when I was a child, and talk a little bit about a passion of mine that I've had since I was a kid. I wrote my first story when I was in 2nd grade, and the story is called The Space Dog. I still have it, and looking back and reading it now, it definitely is not up to standards. But ever since that moment, I knew that writing was a secondary passion of mine. My father saw this and started assigning me graded book reports while I was in high school. At the time, I hated it, but looking back at it now, I was glad that he did because it opened my eyes to a bunch of possibilities, and it really also honed my writing skills. This inspired me to keep writing throughout high school. In a way, I also saw writing as an escape from reality. So knowing that I've always had this passion for writing in my back pocket, I want to talk about kind of where that turning point came in my graduate career. So around year 5 of my graduate career, I felt burnt out. I felt that what I was doing wasn't making an impact in society and in science as a whole. And I've also felt that academia wasn't for me. I didn't see myself in the role of a lab manager. And so I started to think about what I really wanted to do after I finished my PhD. The turning point for me came during a career talk that was hosted by the microbiology department. And at that career talk, one of the careers that was mentioned was science communication. I had no idea that science communication was even a career option for PhDs. And so as soon as I heard that, I became really interested and started to look more into it. I then approached my advisor about it, and she was very supportive and helped me connect with some of her colleagues. And so from that, I joined the science policy group here on campus because I figured, I would have more of an impact if I was able to influence the government. During my short time with the science policy group, I helped organize a writing workshop, which only furthered my interest in science communication because in the workshop, we talked about different forms of science communication. Since I knew that I was invested in this, I actually reached out to a colleague of mine, Ananya Sen, who was already writing a lot at the time, doing a lot of science writing, and she helped me to find writing opportunities for different venues. And so in doing that, I was still finishing up my graduate school, my PhD. And then another friend of mine told me about a fellowship opportunity at the IGB here on campus. And so I decided to apply, send my writing samples, even though I didn't have much experience in science writing. And next thing you know, I was hired and I've been working at the IGB ever since. And so in a way, during that turning point, I went back to my passion. I went back to my interest, which was writing. So in the summer, as I was wrapping up, I decided to reach out to the Illinois Science Council, and they were gracious enough to let me write a piece of a topic of my choosing. And so I decided to write my first science article on the father of the periodic table, since at the time it was the anniversary of that discovery. And this really solidified my interest in science communication and told me that this was the right decision. And so from this, I learned that if you're interested in something, just try it out because you never know until you try. In addition to that science writing article, I also had other writing projects because I wanted to make sure that my mind was clear, my writing was not rusty. And so I tended to like creative writing more, but I was starting to become more interested in science writing. And so during the summer, I reached out to Smile Politely, which is the magazine here in town, and wrote a few articles for them, which was more culture-based. I also wrote some blogs for Black Girl Nerds, which is a space for women to nerd out about anything and everything. Also, I was fortunate to have such a supportive advisor who gave me practice in editing, and so I helped edit an article for the Department of Microbiology. And I will come back to this later, but during that time, I also started writing my first fiction book. So you're probably wondering what exactly science writing is. So science writing is defined as a form of writing designed to communicate scientific information. And this can be in a non-technical manner for a general public or an audience of non-scientists. Science writing can also be for researchers in the area of expertise. Another question that might come up is how my PhD training influenced my science writing. And one of the most important things I think, is scientific credibility and accuracy. Since I am writing for the public, I want people to trust what I write. And so, more than likely, they're going to trust the writing if they see that someone with a PhD background who is an expert in that field wrote that piece. Since I am an expert in, for example, microbiology, I can also represent the science with accuracy and spot mistakes that I might find. During graduate school, I also gained a valuable perspective on the culture of science. And through that experience, I'm able to appreciate the nuances that go into research. As a science writer, we are expected to conduct our own interviews and come up with questions. And so asking the right questions during interviews is very important. As a graduate student, I was able to appreciate research and understand certain concepts and techniques. So I'm better able to formulate questions to bring out the science. Lastly, since graduate school was very technical, I was able to pick up on little details. And for my science writing profession, it's very important that I am able to pick out even the smallest details. So some goals as a science writer, and specifically at the IGB, one of my goals is to help raise awareness of the research taking place at the IGB. A lot of times there's a lot of great research going on at the IGB, but people aren't aware of it or don't know about it. I also realized that there is a communication gap between researchers and the public. And as a basic example, explaining my research to my parents can sometimes be difficult. And they think I work on salmonella when I work on E. coli, even though I've explained, many times. So I realized that this communication gap is something a science writer can help alleviate. Another goal of mine is to facilitate the interactions between the IGB and the greater community. So this can be in the form of volunteering in outreach events or helping provide scientific content for different outreach endeavors. Last but definitely not least, I want to make sure that the science is accessible to the general public, meaning that I'm keeping in mind the audience and also being able to relate the science to their everyday lives. So some skills that I picked up from graduate school that really helped me in my science writing are shown here. And this is not an exhaustive list, but these are definitely some of the skills that I thought were important. First, like I mentioned before, as a science writer, you are expected to conduct your own interviews, and interpersonal communication is a huge part of the job. While in graduate school, we were expected to present seminars, and through that experience, I was able to sharpen my public speaking skills and also being able to interact with faculty, students, and students that I was teaching. So through those years, I was able to develop those communication skills. Another skill that I developed during grad school was editing. And as a grad student, you are busy writing your manuscripts and your thesis, for example. So a lot of editing goes into that process. As a science writer, we edit a lot. And so doing that early on was really helpful and not as hard to get into. Another skill that I picked up was literature searching. So I'm not an expert in everything. So when I come across a new research paper or something that I'm not familiar with, such as physics, I have to conduct a little research and look up articles or reviews based on that topic. As a graduate student, you're constantly reviewing literature. And so this really prepped me for the job. Lastly, I think I forgot about this, but it's just as important. But time management was definitely something that I had to deal with in graduate school because you're conducting research, you're teaching, you're maybe in other organizations. So you had to juggle a lot as a graduate student. And as a science writer, you could be working on multiple writing projects at the same time. So it's important to make sure you juggle the time wisely. So some of the duties as a science writer are listed here. So one of the big things I do as a science writer is write press releases. So the press releases can be about an exciting new paper that just came out from a lab, or a new award or grant that also just came out. I also write articles, and the topics can vary from philanthropy articles to articles about a new upcoming event. And once again, interviews is something that I conduct, and I conduct with my own questions and record and transcribe all the interviews. I also am involved in grant editing services. So some researchers will come to us and will provide us with their grants, and I help edit it. I'm also responsible for social media upkeep. So I help, for example, manage the Twitter page at the IGB. We also have magazines annually that I work closely with the designers on. And lastly, I get miscellaneous requests. And the funnest one that I can think of, or most recent one, was a COVID-themed escape room that we did through the IGB. And this day-to-day variability is what I love most about being a science writer. Okay, so I did say that I would come back to this project, but I will kind of talk about a book that I actually started writing back in 2016, so while I was still a graduate student. So this was the period where I was still trying to figure out what I wanted to do. And during graduate school, I went back to my interests. So I found writing as a welcome escape from graduate school. And during that time, I decided to start a fiction book. And another part of my life that I did not mention before is video gaming. So video games have always been a huge part of my life, and I took some inspiration from that aspect. I also was able to draw some parallels between graduate school and video gaming. One thing being that for both video games and graduate school, I felt that sometimes I was so laser focused on one specific research question that I would forget to look at the bigger picture. The same can be said for a video game where you're fighting the same boss over and over again, and sometimes might forget to think about other options and thinking outside the box. So I framed my book around those ideas, and I saw an opportunity to put my work out there. And so I decided to put my entry, "Are you sure you want to quit?" on a crowdfunding site for books called Inkshares. And if you know Kickstarter, you can think of Inkshares as the Kickstarter for books, where inspiring authors put out their works and hope to get pre-orders. And if the pre-orders meet a certain goal, that book gets full editorial, publishing, and marketing support from Inkshares. Back in 2016, The Nerdist, which is a platform for all nerdy things, they co-hosted a contest with Inkshares. In this video games contest, my book was part of that entry. So Are You Sure You Want to Quit is a story about Maggie, who is a gamer girl who was obsessed with a role-playing game that she has been playing for a long time and can't seem to defeat the final boss, who happens to be a dragon. Through this journey, I play with the idea of being a gamer, but also being a girl in the gaming industry, and also the lines between fiction and reality that begin to blur. I came in 4th place overall in the contest that included 63 entries, and I'm hoping that the manuscript will be done in summer of 2021. So looking back at my journey, I see it as my life coming full circle, where I ultimately started with this passion for writing, and picked up another passion, which happens to be microbiology, and went back to that passion of writing. And now I'm working as a science writer, which combines both those passions. And so I was fortunate enough to find that opportunity. So just some final thoughts. There are many opportunities out there for a postdoctoral career. When I began the program, I thought it was just academia or industry, but I became exposed to so many different opportunities, and I'm glad that I attended that career talk about science communications. It's important that if you are at this stage where you're trying to figure out what you want to do next, to make those connections early and also to take the initiative. I also realized that a lot of the skills that I picked up during my PhD training translated well to science writing. And of course, it's important that you do what you're passionate about. And it might sound cheesy, but it's important, especially to follow your heart. If you know this isn't right for you, maybe this is not what you were meant to do. And so finding those passions is very important. Okay, so I would like to thank, of course, the wonderful Vanderpool lab that I conducted my PhD training in. It was such a great environment to work in, and Carrie is such a supportive and great mentor, and I definitely would not have finished without her guidance and support. And of course, I want to thank the IGB for giving me the position in the first place and for allowing me to explore science communication as a career. Okay, and so with that, I would be happy to take any questions or comments. If you would like to connect or have any other further questions, you can feel free to email me here or you follow me on Twitter. But I will warn you that I'm not as active on Twitter. So, all right, thank you so much for listening.
Yeah, thank you very much, Alisa. Yeah, if anyone has any questions, you can feel free to unmute yourself and ask, or you can put them in the chat. But is that a question there, or is that applause in the? Awesome. So yeah, I guess if we don't end up having any questions, then I want to thank you again, Alisa, for talking, and thank you for helping us out with.
Oh, there's a question.
Yeah, joining us for the Science Cafe. Yeah. What was your biggest challenge for switching from to a writing career from your PhD work?
Yeah, that's a really good question. I think the biggest thing was taking my mind off from such a narrow focus to all of a sudden having to expand my horizons. And, you know, as a writer, you have to be able to adapt and you have to be comfortable with different areas. So it was definitely a challenge for me to get away from the technical aspect. Also, the challenge was switching from graduate school schedule where you just came in on the weekends and conducted experiments whenever to a 9-to-5 job. That took a while for me to adjust, especially because I was so used to coming in late at night and working on the weekends. So I would say those two things were definitely the hardest adjustments. And also, I guess one more thing, I also didn't want to become too technical with my writing since I know so much about my background or my expertise is microbiology. So it was hard for me to let go of some of those details. Yeah, that's a great question.
Yeah, it looks like there was another question. Were there any courses or training opportunities that you took at the UIUC to help you build your science writing skills?
Yeah, so there was a grant writing class, which I actually did not take, but a lot of my colleagues, classmates took the class and they found that very helpful, just to get an idea for what it's like to write a grant or to write a research proposal. And a lot of this, one class that we were all required to take involved reading papers varying from different topics. And so reading papers every week also gave me those literature searching skills, those reading skills that were also important for science writing. Yeah.
I guess were there any resources that you used when you ended up finding the internship or the fellowship at the IGB, or like how do you go about like looking for a job, or how did you go about looking for a job at the IGB or in science writing?
It kind of fell into my lap because my friend who was working at the IGB told me about it. So I discovered it that way. But before that, I was actually looking at job postings. So postings on like Nature Journal, looking at different universities, seeing if they had opportunities for science writing. And I was also looking at science policy opportunities too, because I figured I would get practice in science writing if I got a job in science policy. So it kind of happened for me, I guess it kind of happened. But, you know, if you are actively looking, I would, yes, suggest just starting at those institutions first and seeing if those institutions have opportunities, because I feel like science writing is definitely an important position that institutions need.
We had another question that said, is there a career training path for science communication at UIUC, or did you have to learn on the job type of?
Yeah, I had to learn on the job, and a lot of that came from just knowing the right people. So my friend who had already had a ton of experience science writing, luckily she had all these resources and she was able to connect me to these opportunities. So I basically just kind of went along and did everything. I didn't have a specific track that I followed. So I guess my path is kind of unconventional in a way.
And then another question was if you had any tips on starting to build a science writing portfolio.
Yeah. So yeah, I would first reach out to, like, for example, if you're in like a specific department here on campus, I would reach out to the director of the communications there, see if they have writing opportunities. I know the MCB program, they have undergraduate writing interns. So that is a great way to start building your science writing portfolio. And you can also look for blogs, different blogs, science blogs. They are constantly looking for writers. Like I said, I reached out to the Illinois Science Council, and they're very receptive to me writing something for them. And yeah, even museums, you know, if there's a museum or some other type like that, they are always happy to let you write about one of their exhibits, for example, which I did not know about. But then I looked into it and saw that some museums really like having people write about their historical artifacts or exhibits.
Awesome. Well, we're getting up to 6:30, so if there's no other questions, thank you again, Alisa, for joining us and for telling us about your career. And if anyone has any other questions for Alisa, feel free to message her, or you can message me and I can pass that along. And hopefully you can all join us next month for our next session of Science Cafe.
Thank you.